Okay, for tonight's lesson, we're going to be looking at cell theory and introducing just the beginnings of cells. Okay, first thing I want to do is look at the 7 characteristics of living things. So let's get a basis for what makes something living. Living things grow, they reproduce. If you take them as a group, living things evolve. They're composed of cells. They maintain a stable internal environment. The word for that stable internal environment is homeostasis. They obtain and use material and energy, and living things respond. So I'm going to flip back and forth between these next 2 slides. So what I want you to do is we're going to use 2 different skills. Number 1 is we're going to break down definitions into their most simple form. So I've already done that for you. So for each one of the 7 characteristics, I can break it down into one word. So they grow, reproduce, evolve. They're made of cells, homeostasis, energy, and they respond. Let's go back to this one. So the next skill is turn them into something easy to remember. So what I've done is taken this word Greg chair. So you could turn that into a visual. You could picture somebody named Greg sitting in a chair. That's going to be your way to remember the 7 characteristics. Okay, and then you can play around with that. If that mnemonic works for you, use it. If not, develop another one. So how did we first kind of start to learn about cells? So first of all, Robert Hooke, he looked at cork under a microscope, and that's in the 1660s. He noticed these little compartments, which he named after the little rooms that monks live in, called cells. And next guy that we got is Anton van Leeuwenhoek. And in the 1670s, he looks at pond water, and microscopes have gotten way better. So that's like their technology, their version of the iPod. It gets way better. He looks at pond water and sees all these moving living things. So we see that Hooke sees the basic units of living things, calls them cells. Anton van Leeuwenhoek sees these single-celled organisms swimming around. We see that living things could be 1 cell big. And basically developed 3 concepts for cell theory. Every living thing is made up of 1 or more cells. Cells carry out the functions needed to support life, and cells come from only other living cells. And I got a little visual for you. So concept 1, polar bear is made up of many cells. Concept 2, different cells in a polar bear's body do different jobs. And concept 3, all polar bear cells came from a single living cell. So the polar bear baby came from its parents. What specifically? From cells from a mother and father polar bear that came to make that baby polar bear. So let's break this down. So how a cell becomes a cell. So to make a cell, you need an outside and you need an inside. So the very first thing that probably would have formed that was like the precursor to a cell was a lipid kind of bubble or some sort of lipid container that could form the inside of a cell. And this lipid barrier allows conditions inside to be different than those outside. And that's kind of the very first component of a cell, is you have this inside and outside conditions. Then once you have the inside conditions that you can keep differently, you could add other things in, like genetic material, so DNA or RNA, and you could add in proteins, and we've already talked about enzymes, so you could add in these things that will allow for reproduction and other cell functions. So if you want to kind of get a little bit more out of this, you can look, if you just Google search the first cell in Discover magazine, there's a kind of a cool article about scientists researching how the very first cells could have come to be. So we're going to continue with that theme of the kind of the outer part. We're going to start with the most basic part, which is the cell membrane. That would have been the initial thing forming. So it's a thin, flexible barrier around the cell. The name for the lipid barrier is a phospholipid bilayer. It's just because that this thing over here has phosphorus in it, so that's how it gets the phospho part, and lipid, because it's got this little tail here. That's just like the lipids that you studied in the macromolecule lab. The phosphorus part really likes water. The lipid part hates water. And so that basic hydrophobic and hydrophilic part is what's going to, that basic kind of underlying component of the cell membrane is what's going to allow it to set itself up. So the hydrophilic part's going to plug into water because it likes it. The hydrophobic part's going to orientate itself away from water. And that's going to, so this part here, with the little hydrophilic parts, and this part here, this could possibly be the outside of the cell, and everything under here could be the inside. And then you got this hydrophobic part is your barrier in between those two parts. So the functions of your cell membrane are going to regulate what enters and leaves the cell, and provide protection and support. So all cells have to have a membrane. Whether it's a plant cell, animal cell, or bacteria, they need to have a membrane. Next thing that you're going to do with the cell is you got to put something inside of it. So your internal conditions, you've got to have something in there. That something is cytoplasm. It's a kind of like a jelly-like material that's inside the cell. There's lots of chemicals and proteins within the cytoplasm. All cells also have cytoplasm. And I have this nice little simple picture of a cell here, and that's the cytoplasm inside. Cytoplasm is also going to provide support for the organelles in there, kind of act as a cushion. That way if the cell gets bumped, it doesn't destroy the internal parts of it. It's got that cushion to absorb shock. So you can't just have this enclosed environment with nothing coming in or out. Then it wouldn't be a living thing. So we already talked about living things needing to respond. So a cell also needs to respond. It's got to release waste, and it's got to take in resources. But how does that happen? So there's a couple different ways that cells can transport things. The basic, the underlying idea for movement of things in and out of any movement of molecules is going to be based on a principle of diffusion. So with diffusion, molecules are going to move from high concentration to low concentration. The example that we have here is if you put a lump of sugar in water, the sugar lump is high concentration. It's just this packed sugar. When it goes into the water, it dissolves and it spreads throughout that liquid. So you don't have to stir the lump of sugar necessarily to get sugar molecules to distribute themselves throughout the water. They're going to move because of diffusion. If we dropped a piece of dye, an ink, or a drop of ink in water, that drop of ink or the dye is going to spread over time because of diffusion. Or if the person next to you is wearing perfume or cologne, you can smell them because the high concentrated perfume or cologne is dispersing away from their neck from high concentration to low concentration. So that's what causes molecules to move. And then how they move throughout cells. So they've got to, we know that molecules are going to be moving, and we know that we have this container that's keeping internal and external conditions different. So one way that materials move is facilitated diffusion. So facilitated is like helped. If I'm facilitating your learning, I'm helping your learning. So that just means diffusion with a helper. And that helper could be a channel protein or a carrier protein. And it's just going to allow molecules to move from one side of the cell to the other. They're still going to move from high concentration to low concentration. They just need this like channel or molecule to help pass them along. It's kind of like a gate in a city wall. These gates help to pass larger molecules through the cell membrane. So, and the name again was facilitated diffusion. Are you still awake and alive? Here's a picture of a Jesus Christ lizard using surface tension to escape a snake. Okay, so how could a cell move? How how can we move, what happens if a cell wants to move things the opposite direction? So if it wants to move molecules against their normal diffusion, it's got to use active transport. So the only way to do that is by pumping energy into some sort of protein channel that can pump molecules in a direction, kind of like a sump pump pumping water out of a basement. You want to move molecules to a different area. You want to move them against the concentration gradient. So if you want to move them, actually this should say, yes, it should say low concentration to high concentration is right. So in our example here, we have hydrogen, and we're pumping hydrogen across outside of the cell membrane, and this little ATP is representing energy to do that. We have a special name for the diffusion of water across a membrane. That's called osmosis. So we're going to be able to move water. You're going to be able to move water, or water is going to be able to move through a cell. So you could set this up. Let's say we had a glass, and we set up this semi-permeable membrane. If you have a high concentration of sugar, and sugar is the red, and you have, which means that sugar is taking up most space, water is only taking up a little bit, and then on the other side of our membrane we have a lot of water molecules and just a little bit of sugar diluted. Water is going to move from high concentration, which was over here, to low concentration. So water, eventually the number of water molecules on the left is going to be the same as the number on the right, but we also have all this added sugar, and so our volume would change. The volume would go up on this side and down on this side. So this is like our, I don't know, our lab setup example to look at. Another thing you could think about would be someone drinking salt water. So if you were in the ocean and you were in a boat and you just drank salt water, the salt concentration of the seawater is going to pull water out of your cells and cause you to be more dehydrated than if you just drank regular tap water. So that's why you can't, if you're stranded in a boat, you can't drink seawater. Is because it's going to pull water from your cells and dehydrate you. Or you could think of if you watered a plant with salt water, that salt water would pull water from through the roots, pull water out of the plant, and the plant would eventually die because it would not have a lot of water. Okay, so what kind of conditions does this create in the cell? There's basically 3 different things that are going to happen. In a hypotonic solution, you've got more water concentrated outside. So we just learned, because of osmosis, water goes from high concentration to low concentration. So it would move across and into the cell. At top, we've got in our example, it's like a red blood cell or an animal cell. It would move into that animal cell and cause it to explode. So PO is just like explode, hypotonic. In a plant cell, at the bottom, it just moves into the cell and then fills the cell up. Because of the cell wall around a plant cell, it's just going to like fill the cell up and cause it to be like, you know, a happy plant cell. In isotonic, isotonic is balanced. That means the concentration of water on the outside and the inside of the cell is the same. So water is not going to move in or out. It's just going to move equally in both directions. Well, it's not going to move more in or more out. It's going to move equally in both directions. And so this is going to be balanced. The same with our plant cell. It's just going to be balanced. And then the final example we have is hypertonic. Hypertonic is when you have more water concentrated inside the cell than out. Water is going to leave this animal cell and it's going to shrivel and shrink up. If it was a plant cell, same thing. The plant cell would shrivel and shrink up. In a plant, if it's in a hypertonic solution, it's going to wilt. A hypertonic solution example for an animal cell would be drinking the seawater. The water has left the cells and is gone out in the cell shrivel. Bam.